Sequencing of multivirulent and multiple drug-resistant staphylococcus strains isolated from acne patients. This project is presented by Arij Ayman, Mariam Nagih, Myrna Ahmed, and Suad Abdullah, under supervision of Associate Professor Dr. Hibatullah Magdi, Dr. Amani, and Dr. Nurhan. The main points that we will discuss today are aims, objectives, introduction, phylogenetic tree, methodology, results, and reference. Aim of work: determine the phylogenetic tree of antibiotic-resistant strong biofilm-producing Staphylococcus epidermidis isolated from problematic skin based on whole genome sequencing. The objectives are determination of strong biofilm-producing isolates and which of them is antibiotic-resistant, and determination of their phylogenetic tree to help find a proper drug in the future.
Hello everyone, it's Suad and I'm going to start with the introduction. Hello everyone. Acne is a skin condition that's very common in teenagers and middle-aged individuals, in which the hair follicles become plugged with oils and dead skin cells. It may appear as a blackheads, whiteheads, or maybe pimples that full of pus and inflamed. It may not seem that dangerous physically, but it can leave a very bad impact mentally on the patient if left untreated, especially if it cannot be cured. It can lead to depression. There are several factors that can cause the acne, and one of them is bacterial infection. The most common species of bacteria that can cause the acne are Staphylococcus species. According to a study done last year, they collected samples from both healthy and acne patients, and the bacterial isolation process showed that the most common species of Staphylococcus that causes the infection is Staphylococcus epidermidis. Generally, the Staphylococcus strains are gram-positive cocci, facultative anaerobes that arrange in grape-like clusters, and it's present as a part of our normal flora. According to the ability of the bacterial isolates to produce biofilm, they are classified into non-biofilm producing, weak biofilm producing, moderate biofilm producing, and strong biofilm producing.
And now I'm going to talk about biofilm formation and antibiotic resistance. Biofilm is a surface made when bacterial communities attach together using polysaccharides and self-produced polymer matrix. Then it attaches to the surface of the infected area, forming a protective layer. Furthermore, bacteria becomes able to adapt to poor environmental conditions. As a result, bacteria become capable to resist antibiotic effect and survive. Phylogenetic tree. It's a diagram representing the gradual development relationship between living organisms, and it shows the development of the organisms from a common ancestor to a different species. It's important as to reconstruct the tree-like pattern that describes the evolutionary relationships between the studied organisms and the most common ones.
Hello everyone, I'm Myrna Halima, and now we're moving to the methodology. First, mitosis agar identification test. Isolates were cultured on MSA media using stricken technique and incubated for 24 hours at 37 Celsius degree. After incubation period, Staphylococcus epidermidis grows as red colonies without any change in media color, and the chosen isolates were cultured and their colonies were sent for multi-self identification and 57357 organization to confirm their identification. Then, a biofilm production. The isolates were cultured on TSA media using stricken techniques and incubated for 24 hours at 37 Celsius degree. Colonies were added to saline using sterile loops and adjusted against 0.5 microliter solution. 100 microliters added to 10 ml TSP broth, and then added to a microtiter plate using 3 replicates for each sample and 1 replicate for negative control, and incubate for 24 hours at 37 Celsius degree. Then the replicates were read in ELISA reader at 600 nanometers and 200 microliters of 0.1 crystal violets added to the microtiter plate and left for 15 minutes. Then the stains removed and cells washed out with distilled water and then left to dry for 1 hour. At 200 microliters of 33% acetic acid is added to the cells, and then again read in the ELISA reader at 545 nanometers. Then the results are calculated using biofilm formation index equation to classify isolates according to their biofilm production ability and know which isolates are strong biofilm producing. Then we'll move into the results of the biofilm production test. 23 samples out of the 60 were strong biofilm producing Staphylococcus epidermidis isolates. Which represent 38.3% of all the isolates, and 6.7 were moderate biofilm producing, 6.7 were weak biofilm producing, and 48.3% was biofilm non-producing.
Finally, let's move on to sequencing and phylogenetic analysis steps. 4 isolates were chosen according to their biofilm results based on their strong biofilm formation ability and antibiotic sensitivity. Then, the selected isolates were sent to 57357 hospital for sequencing and phylogenetic analysis. The phylogenetic tree is not known yet as the sequencing step is still in processing. This is some of the references that we use to collect our data. Thank you for listening to us, and we're welcoming to answer any of your questions.